Hello, everyone. Welcome to this new seminar. So today we have Luisa presenting, and I'll let Ignacio introduce today's speaker.
Okay. Thank you. Well, I'm extremely happy to introduce Luisa Pallares, to have host Luisa Pallares here. Luisa is an evolutionary biologist after Luisa training in Colombia, and then obtained a PhD in Plön with Dita Taut on evolutionary genomics, then moving to Princeton to work on G by E interactions, and then since 2022, she is a group leader at the Max Planck Institute in Tübingen, working with a strong emphasis in the last years on the role of stochasticity and noise in biology. So I'm truly happy to have you here, please, Luisa.
Thank you, Nacho, for the introduction and for the invitation. Thank you for everyone who is here today. So yes, I will tell you today a little bit about what we are doing now in the lab, which focuses on phenotypic variability. But because I've given this talk a couple of times and people always disagree with the terminology I use, I thought we're going to start from the beginning just to make sure that we are all on the same page about what we are talking about today. So what we do in the lab, in very general terms, is trying to understand why individuals look different from each other in a population. So all studies we do with Drosophila melanogaster is population-based inference on variation between individuals. And we are particularly interested in understanding how this variation, this phenotypic variation at the population level, is driven or influenced by genetic information. So we are really focusing on this part of the equation, is trying to understand how this information propagates to between individual variation in the population. And then how do we do this? This might be very basic for probably most of you, but just in case. The way we think about trying to fill these arrows here is using population level inference of genetic association between phenotype and genotype, which is GWAS, right? So this is a Manhattan plot, which is the result of a GWAS study. And the way we have tried to approach this is, ancient history, but I always like an excuse to show what I used to do before in my previous life. When I worked with mice, is what I did in Plön before transitioning to flies now. So, but back then and even now, what we try to understand is why individuals look different in the craniofacial shape, for example, or in the levels of gene expression. And what we do basically is we walk through the genome of whatever is your organism of interest, and we try to find significant associations between your trait and the genome, right? Just to make it more explicit, what we do when we find a significant association between your phenotype and your genotype is that we are looking for this. Basically, we are running a linear regression and we're trying to see, okay, the phenotype changes depending on your genotype. So this is a position in the genome that we're interested in, and then we try to understand this genetic architecture. But I just want to bring your attention to, for example, some positions in the genome that you will never consider, you will ignore and never think about them. Because when you do this inference based on regression, this genotype in particular, in this chromosome in particular, has no correlation with your phenotype, right? So by definition, it's not a locus that you're interested in because it doesn't affect your phenotype, right? The fact that this is the way we try to understand genotype-phenotype relationships has a consequence, and we can discuss this during the discussion if you're interested in whether this is more biology or more a product of how we make inference, is that we used to think about genotype-phenotype maps in a deterministic way, right? We try to understand the variation in this position of the genome, and we imagine that if you have a C, then you tend to be orange, and if you have an A, you tend to be blue. And I mean this in a deterministic way because the inference we're making is about the average of these distributions, right? Is that meaningful? Is that a good way of approaching phenotypes? I hope we can talk about that later. But going back to this actual, it's a toy example, but it's the real one at the same time. I hope you appreciate that for every genotype that we're trying to map, there's a distribution of all the data points that you're mapping, right? Even in this example, I hope it's very clear that these GG guys are super widely spread compared to what is going on in the CC genotype, right? And then this is what I'm going to be talking about today, and I'm calling it variability in this particular context, but you will see that I will lose my rigorosity quickly and call this many other different ways that I will tell you about it in a second. But this is what I want you to think about, right? It's what's happening with this spread of individuals that we normally ignore, either because our sample size is not good enough to make inference. We don't know if it is noise, if it is technical error, we don't know. But I just want to bring your attention that these things that go beyond the average of your estimate might be very interesting. That's what the lab is focusing on right now. So many people have thought about going beyond averages when I'm thinking about phenotypic variation. And this is just some of the examples of fields that have made a lot of progress in this particular topic. So people working in morphological structures have been approaching this idea of developmental robustness or the variability that comes from noise in a developmental process by using what we call fluctuating asymmetry, which is the comparison between right and left sides of symmetrical individuals. Just because it's the same genome making your right and your left side, the null expectation is that you should be symmetric, right? Deviations from there is developmental noise, for example. People thinking about genoplasticity have been thinking a lot also about this idea of multiple phenotypes, so not only about the average phenotype that a genotype can generate. The field of transcriptional noise, for example, has been going on for decades, where they have focused on if you have one genotype, one cell, control everything, why do you still have variability, right? And they try to approach the stochasticity that generates this transcriptional noise. Even people thinking about quangen have been trying to model the heritability of this variability, which is non-zero, right? So there's genetic information that regulates how widespread beyond the mean your traits are. And more recently, I would say, people who are trying to study behavior, right? They study individuality, even now in cells, but also in individuals, is this idea that within a genotype, these are two different genotypes, whatever behavior you're measuring has also differential spread between genotypes. All this evidence has suggests that, yeah, this noise, okay, this is important. I will use robustness, I will use noise, I will use variance, I will use variability. All this means kind of display, right? This noise has a genetic component, and therefore we should be paying attention to it. So today, I'm going to tell you first a story about transcriptional noise, which is a thing that we have been thinking about for a while already, and later on a little second story. So when you think about this variability, about this variance, I think a reasonable question to ask is whether these things are evolutionarily relevant, right? They might be there because stochasticity is there in every biological system, but doesn't mean something that is interesting to evolutionary biologists, right? So this project was led by Scott and Diogo when I was at a postdoc back in Princeton. And the way we tried to put this question was, if transcriptional variability is a gene property, then it should be constrained by evolutionary and functional properties of how genes evolve, right? And then the assumption here was, and I always make an emphasis on, for people here who have worked with gene expression, these are really noisy metric, right? Like if you have batches, if you don't design your experiment correctly, there's like so many things that are affecting gene expression, that if you say you want to do the most tricky experiment ever is try to measure if noise in gene expression is actually something important or just technical noise of the technician or the student who collected the data. So we said, okay, let's push this and try to figure out if this thing makes any sense in a population, I'll tell you in a second, that has genetic variation, right? So this is not within genotype, but just in normal population. So what we assumed was the following. If transcriptional variability is a gene property and I have the same gene measuring Nigel's lab or measuring my lab by different students and different technicians, so all the noise technical that you can add to the experiment is there. If it is a gene property, it doesn't really matter where you measure gene expression of this gene. The variability that you're going to measure is going to be highly correlated between your locations, for example, right? So if this is true, and this axis is every single dot is a gene in your genome, this is the variance of your gene expression in my lab, this is the variance of gene expression in Nacho's lab. If our assumption is correct, and this is actually a gene property, you're going to have a pretty nice correlation where genes that are low variance in one condition and low variance, doesn't matter where you measure them. And if you have a super highly variable gene, it will remain so, doesn't matter what you measure. Does it make sense? If this is the null of this assumption, is that this is just technically induced by whatever you're doing, and therefore if you are comparing across conditions, then you should have a no correlation between this particular metric, which is variance, right? Mean always behaves like that. It's a really, really robust metric, and the mean of genes is really reasonably correlated. So what we did for this is we said, okay, we need as many studies as we can have. So we can do this pairwise correlation in as many conditions as we can. So we went for human data, because that's where you have tons of these studies done in whatever tissue you want, whatever lab you want, in sample sizes that are reasonable to be able to estimate variance, right? Which is also very important. So we find around 57 studies. After you overlap everything that you can find in all of them, you have 65% of the transcriptome represented, which means you can make reasonable inference. And then, spoil the result. And then we use, this we can discuss later if it's important, you have to be able to decouple mean and variance correlations that happen always, not only in expression, but in every trait that you're measuring, to be able to figure out what's going on with the variance. What did we do was just to basically do pairwise 57 against 57 ones of these particular plots to see what was the trend, right? And then, long story short, this is the result we found. This is the distribution of these strengths of this correlation across all these pairwise comparisons. And I hope, I mean, in biology, high correlations are hard, but the gene expression is really tough. And most of the correlations are really, really strong granular correlations beyond 45%, right? So this means genes that tend to be noisy in one are going to be noisy in the next. Okay, so if this is true with a gene property, since that is reasonably conserved across technical noise, now the question is, what is constraining this variation across genes, right? So what we did is we took the bottom 20% of genes that are super highly tightly regulated, so there's not a lot of variation, and the top 20% noisier genes in the human transcriptome, right? And then we ask, what do these genes have in common, right? So we run correlations across these groups, and we find something that, tell me if you're surprised, but for us, was like this kind of makes sense. And these genes that are very, very noisy can really accommodate a lot of genetic variation in their sequence and in their regulation. So when you are very, very tightly regulated and always being expressed in the same way, you actually have very little genetic variation in your sequence. When you are very conserved and not noisy, you are very intolerant to perturbation. So these genes are like, you cannot touch them. In humans, there's this database, and when you perturb these genes, you cause disease or the human doesn't develop. And these genes are super intolerant to any mutations that you have. So they are very, very important. These guys are fine. You know, you can mutate them and they are happy. Then connectivity in gene networks is a very interesting thing that has been explored also for other systems. And these genes that are really reliable are super well connected. So they are hubs in your gene expression, in your gene co-expression networks. Well, if you're this crazy person that today is happy tomorrow is mad, I don't know, you're not very reliable friend, maybe, then you tend to be in the periphery. You don't have many, many friends in these gene co-expression networks, which I guess kind of makes sense, right? And then finally is, what are these genes? You know, as biologists, we always love to do geo-enrichments to try to see what these things are doing. So then we did it for top and bottom variability genes. What do you think these genes are enriched for? A group of genes that is super reliable and a gene that is like whatever. What do you think they are functionally enriched for? Any guesses?
Information in the document or
Information. What thoughts? Fox genes.
all the core biological processes left cell cycle.
Exactly. So all the things that are like super fundamental for a cell or for a system to work, doing them indeed are enriched in super highly reliable genes. So these are all housekeeping. If we want to call information transfer, for example, to translational process or transcriptional process, all this is really, really very uniquely enriching this group of genes. So you don't get like 20 million things enriched there, but these particular processes. On the other side, the highly variable genes tend to be things that respond to environment or are related to new processes. Are we surprised? No, I think all this kind of makes sense, right? So this is really cool to see that there are all these forces, evolutionary and functional, that are shaping the amount of stochasticity that certain genes can express in a population, right? And this is just to highlight that this is a study that we did in a population that has genetic variation, right? And these questions have been explored before by Sandra, for example, in Arabidopsis, but also by people, as I mentioned before, that have used single cell unique genotypes, right? So in these studies, there's no genetic variation. We're trying to understand stochasticity of gene expression. What we try to do is, if we go and work with populations like the ones we work, for example, with Drosophila, with humans, do these things still survive, right? Our interpretation of this is that you have this higher order constraint on the amount of stochasticity that a gene can express, and then genetics comes and fine tunes this amount of variation that you can have, and this is the second part that I'm going to tell you. Okay, so because we're evolutionary biologists and we like to think a lot about adaptation and what happens if I put you in a new environment, what's going to help you survive or die, we thought, okay, this thing seems to be very interesting, transcriptional variability. Now what happens in a new environment, right? Like if I expose my system to new conditions, is this a trait that responds to environmental perturbation, right? So here you have to go to an experimental system, and now we start talking about Drosophila. Just want to tell you a little bit about how we do it in the lab. So because we're interested in variation between individuals, we work with populations that have variation between individuals at the genetic level. So we are not working with inbred lines, which is normally what you will do when you are doing exploration of Drosophila experiments, but we're working with what is called outbred population, which means every single fly in your population is like a human, has this unique genome. And this gives you a lot of advantages if you are interested in the genetic basis of whatever, because then you can do very powerful mapping for these particular traits. So the populations we have in the lab were collected in Andy Clark's lab a long time ago, covering all these regions, geographical different regions. And what we did is we recombined the inbred lines that were collected in these locations for hundreds of generations to make sure that they are randomly mating and that the LD blocks are very small. And at the end, we end up with cases like this one. So these are the normal bios that probably you have seen, people working with Drosophila. We keep our population in these big cages because you want random mating to keep this genetic variation there. So these are, we like to think closer to situations that they are outside where they can fight, fly, escape, or do a little bit of more normal things. And these are the populations that we use to do this mapping that I'm going to tell you about in a second. Okay. So how do we do this experiment to test what transcription variability does in new environments? It's a project of James, a PhD student in the lab. So what James did is took one of these populations that has this big population size in outbred setup, and he exposed the flies to control and to high sugar diet. Because high sugar, they don't like it. Flies are very stressed. They really get obese. They get insulin-related symptoms. They're almost diabetes. They're used as a model for diabetes in humans, for example. So we know this is a stressful environment, and we are placing them for the first time. So we want to see what happens if you stress these populations. What happens later is that we let them develop for a whole developmental cycle. So what I'm going to tell you about are adults that developed the whole time in these different diets, right? So they every single step of the development of these flies was exposed to this diet. And at the end, we collected around 2,000 samples that have whole genome sequences and whole transcriptome sequences. And this is only possible because we suffered through developing this protocol that allows us to collect from single flies genomes and transcriptome for very, very, very cheap. So it's a couple of euros compared to you do it with a kit, it will be just impossible to collect these sample sizes. And therefore, we have a data set, as I mentioned before, that has a good statistical power to be able to catch differences in variability. This is just to show you that when you're trying to make inference of what a genetic pool does in two different environments, you have to make sure that it's not genetically structured, right? Like if the genetic composition of your two environments is different, then your inference is on that structure that you are seeing. This is a principal component in red, high sugar, and in gray control. And as you can see that the genetic compositions, this is coming from around 300,000 SNPs. It's pretty much the same. You don't see structure between diets, right? So this is basically the same parents lay eggs equally in two conditions. So you have the same genetic pool represented in the next generation. And this one is a principal component of the gene expression data, just to show you that putting them in high sugar does change the transcriptome, right? But we are not going to be talking about how the mean of gene expression changes, which is what the PCA of mean expression will do. We are trying to figure out what happens with the variance of this gene expression. So the first thing we did was to run a differential variability test, which is, the cousin or differential expression test. And instead of checking if the average amount of the gene is different, you're checking if the variance is different given your environment, right? So this is just to show you that this is insane. Almost the whole transcriptome changes in variance once you're stressing these populations, okay? And this is a volcano plot for variance. Everything that is to the right are genes that increase their expression variability in high sugar. And the ones that are below zero are the ones that decrease gene expression variability. So this is just to show you that if you want to imagine something more cartoonish, this is the scenario where we are living, and the control happy conditions. Genes have whatever average level of noise, but when you stress them, all the whole transcriptome increases the level of noise. Okay? Because having a baseline is always super important. This is what happens if you do differential mean expression. Just to show you that around 50% of the genes change in expression, almost half increase, almost half decrease, while almost 90% of the genes change variance, right? So mean and variance are changing different, and it seems that perturbation is changing variance in a stronger way than it changes mean expression. Okay. And then we said, well, the whole transcriptome is increasing variance. Is that interesting or not? I mean, if you have everything changed, what can you get out of that? Right? Like if I run an enrichment on this, then you get everything. And then we thought maybe more interesting to follow the patterns that are going against what the whole transcriptome is doing, right? And then you have a couple of genes here that is really, like, I forgot, very, very little in percentage of genes that are actually tightening the regulation. They have less variation between individuals once they are stressed, right? And we say, okay, this looks like a very interesting thing. What's going on there? When you do geo-enrichment on these genes, I'm coloring them black when it's only change in variance. So mean stays the same. I love this example because there's no, this statistically is decoupled, but biologically is not, right? But these genes don't change in mean. They don't care, but they reduce variance. The orange dots are genes that reduce variance and change mean, but the mean can be higher or low because money goes everywhere, right? The consistent thing is that they reduce variance. And this is brilliant. I think we were very excited when we found this enrichment because it's, again, very specific. It's not that you get 20 million geoterms and what do you do with that? It seems that it's really development and metamorphosis in particular, the pathway that is somehow acquiring stronger regulation, or you don't want to vary as much as you did when you were happy, if you pardon my language, in the sense of intention, right? And in neurogenesis in particular, it's also doing the same. And now is a $1 million question is how do you link reduction in noise to higher order phenotype, right? So this is suggestive that development is something that being tightly controlled in this situation. How do you do it, right? If you have ideas, tell me. But then what we thought that we could do is to at least go and measure development and see what's going on at the developmental level, right? So what Saudat, who is another PhD student in the lab, did is she measured the classics, and it's how long does metamorphosis take in high sugar and controlled conditions. And it is, indeed, this was known before, 20% lower in high sugar. So it takes the flies almost 1 extra day to become adults. So something's going on. They are stressed, they are handling a new environment, and they are super slow. Normally we associate this with, this is a problem. But if you go and measure the survival, so the amount of adults that come out of the egg, so the success rate of your metamorphosis is actually better in high sugar conditions. So they are struggling, but the QC, the quality control they are doing in this metamorphosis process is better. So they have more adults out of the same number of eggs that a control batch would have. And we think this is, of course, the most non-causal association that you can do, but I think it hints that something is going on that is very interesting at the developmental level. Okay, and now we get to business. All this is happening. I have a super strong phenotypic response when I expose these things to stress. Now, is this genetically regulated or is this whatever, right? Just about, I don't know, the system is freaking out and everything is changing. So what do we do for trying to find these genotype-phenotype associations? We did a GWAS. And this is just to show you here that another advantage of doing mapping with outbreak population is that basically you are limited by the number of individuals you can sequence. When you are working with inbreds, you are limited by the number of individuals that exist, okay? And this is just a recent publication where we estimated the regulation for mean expression, not variance. This is just a saturation curve to show you that this little red arrow is the number of genes for which you can find EQTLs with the current panels. And this is what you can find if you can actually increase sample size as much as you can afford, basically, right? Just to tell you that if you have these things, and you can sequence a little bit more than 120 or something lines that we have now, then you can really, really start discovering a lot of what's going on regulation-wise. So therefore, we keep using these flies. This is just a reminder that we have this set of around 2,000 genotype transcriptome pairs, individuals, so we can do this type of association. Just to make sure that we are thinking about the same thing, this is what an EQTL mapping would do. There's an association between your genotype and the average expression of your gene. And this is what a V EQTL mapping does, or a variance EQTL mapping does, is looks for a relationship between your genotype and the spread, the width of this particular genotype. We don't care. We do care because we account for it in the model, but we basically make all the changes in mean homogeneous. So this is not driven by mean. It's just driven by whether the variance is regulated. So James did EQTL mapping, and we were super, super happy because we found that there's pervasive genetic regulation for noise. There's, at least in part, tons of information in the genome that are regulating this stochasticity that we could think is stochasticity, right? Like, I mean, what does it mean that this thing can be spread? No, there's something in this AA genotype that makes this spread possible, and something in the GT that reconstructs it, right? So we find that this is true for almost all the genes in the transcriptome, and I want to highlight something very interesting. This is, again, mean, and we find genetic regulation for mean for almost all genes. And we find regulation for noise for almost all genes. This is kind of known because we have been doing EQTL for a long time in many systems, but this is kind of new. It's like now this additional parameter of gene expression or phenotypic variation is also genetically regulated. But to me, the coolest part of this result is that this genetic regulation seems to be unmasked only when you're stressed. So if you pay attention to this plot, this is the number of genes that you have regulation for. This is most of the genes that have regulation for the mean. You can identify these statistics, right? You can find these genes in control and in high sugar. So this genetic regulation, doesn't matter if you're happy or stressed, is there. But when you come to the VEQTL, we can only detect this genetic regulation under the stressful condition, right? There are some of them, of course, that are shared between control and high sugar, but most of the action in regulating variance comes when these populations are stressed, right? And this is what we call cryptic genetic variation. It's a flavor type of environment interactions. When if you know the yellow and you imagine that control conditions are these gray lines, is irrelevant. It doesn't matter if you have a G or C, the spread, the variance of your gene expression is the same. Once you put this GC in yellow, which is high sugar, now this G guy goes crazy and then the variance increases. So it's cryptic variation because you have the same genetic variation that is hidden in one condition and exposed in the second condition. Good. And then we asked, is high variability beneficial or detrimental, right? And I want to know the answer, which means I don't have an answer for you, but we try to get an answer for it. And do let me know what do you think about the type of inference we try to make. Because this is extremely interesting if you think about it, right? We have been thinking about noise in biology for forever, and we can debate here whether people think it's good or it's bad. There are tons of scenarios where high variability would be beneficial. There's tons of scenarios if you're in the fitness peak of if we assume most of the things are under stabilizing selection, then you don't want to have individuals that are making you fall from your fitness peak, right? So noise maybe is not what I want. If you are adapting to new environments, if you think about plasticity, bed heading, or whatever, maybe having high noise is beneficial. So this is, who knows, right? It really depends. So we ask this question because now we have thousands of positions in the genome that regulate this noise, right? So now we have something that we didn't have before, right? So we can make inference and models of what happens if your phenotype is noisy or not, but now we know where they are, we know who they are, and we can ask what do they do? Can we infer if they're good or bad? So the assumption here, and this is again, I think the best you can do with this type of data, because to be able to answer this, you need to quantify noise and fitness somehow in your individuals to be able to say if the individuals with high noise are more fit, right? We don't have the data. So what we thought was the following. If we base our inference on derived alleles, we can ask if derived alleles are biased toward high or low noise. And this is under the assumption that all the genetic variation that comes new in a population will tend to be deleterious, slightly deleterious, right? So we say, okay, if most of this variation is going to be detrimental, and this detrimental variation has a bias in high or low noise, then we could assume that then this type of bias, low or high noise, is detrimental, Does it make sense? How do we do this? You have to infer the identity of your alleles, whether it's ancestral or derived, first of all. So what we do is we take every position in the genome that we have SNP for, and we use a phylogenetic Drosophila that has 3 species, Jacoba, Sibilans, and Melanogaster. And we determine, for example, if CC is an ancestral allele because it's present in all the species, or if it is a derived allele because it's only present in Melanogaster. Did I mention that all we do is with Melanogaster? That's important. This is our experimental population, right? So that's why this is our focus, and we compare it to 2 other populations. So after we do this, we're able to polarize around half of the SNPs that we have, so around 150,000 or so. And why am I bringing EQTL here? I hope this makes sense. There's no reason that I can come up with that will say that having higher expression or lower expression is universally beneficial or detrimental, right? You will have high expression when you need it, low expression in different gene or whatever. So there's no assumption, there's no expectation for this to be biased. But as I said before, we could discuss there are many assumptions in which, in many situations in which I can think that having high-low noise, if you imagine this is really far away from the average and you are in a peak, it could be detrimental, right? So there are ways of imagining how a bias here makes sense. I don't expect any bias, universal bias, when you have high or low expression. So this is a really cool thing that we can do with this data set because we can actually not only make inference on VEQTL, but we can compare it to something that we assume and understand better, that is EQTL, where we don't expect this type of things to. So these are the results for EQTL. This is the fraction of these derived alleles that decrease noise. So these are low noise alleles. This is another distribution that we create with all the background SNPs that are not EQTL. So we assume this is the random distribution of what you can measure. And you see that the bias in EQTL is not there. It's really, really tiny. So it's really, there's no bias for low or high expression levels in EQTLs. But when you do this for VEQTL, then the bias is huge. This is again the noise distribution. And then this is telling you that when you are a new allele that arrives in the population, you tend to reduce noise. So if you're following me, this means that these derived alleles tend to be low noise. Therefore, low noise it's suggestive of having detrimental effects in this population, right? Which is very interesting if you ask me. If you have asked me, I would have thought maybe the opposite will be what will come up. We can discuss that after. So we're really, really very excited because I think this is, I mean, how else are you going to test this? I mean, you can, but then you need a whole different experiment. But this is really, to us, a really interesting signal that low noise, evolutionarily speaking, might be, in general, not what you want to do. Okay, all good until here? Okay, so now we move to a different level of organization in an individual, right? So every inference we make until now, it was based on transcriptional noise. And we are really very excited because I think what we are getting there is that phenotypic variance at the transcriptional level, it's a trait on its own. It seems to have a set of genetic regulation that is distinct from what regulates the mean, not only in where these loci are, I'm going to talk about that, but I can tell you later the details, but also the selection pressure that these loci might be subjected to. So it is a different trait that behaves with its own properties compared to the mean, right? And then the question is this something that we can generalize? Is this transcriptional only? Is this something that happens across the genotype-phenotype map if I measure all the phenotypes, right? So what we thought about doing is, okay, let's focus on morphological variation because you can infer not only the variability in this particular trait, but because if you have symmetric structures, you can actually infer noise per se, like true noise at the developmental level, right? So you kind of can get multiple phenotypes out of one, and I will walk you through it in a second. So the question was basically, what is the genetic architecture of variability in higher order phenotypes, right? Like, do we find the same patterns that we're finding in gene expression? And this is a project that Saudat led in the lab. And what she thought was about collecting individual level phenotypes plus genomes in Drosophila, right? So what she did is collect around 1,000 flies, going from top to the bottom, where she collected functional behavior. So she measured how good these flies fly as a function of this individual at the individual level. And for each individual, she collected measurements of the morphology of the wings, because the wings help you fly, so this should be somehow traits that are correlated. And then, because you are measuring a symmetric structure from the same phenotype, you can infer developmental robustness at the individual level. And then at the end, you have suffered through all of this, then you dump them in a plate and you do DNA extraction, and then you can do genotype phenotype mapping for this phenotype. So how do you do this? This is basically, we're discussing a little bit about that today. It's just multivariate statistics of how you are making inference on these phenotypes. What you do is you use geometric morphometrics on the wing of your flies, and basically you put landmarks that at the end are XY coordinates that you use as a vector to make inference from there and onwards, right? If you do this in both wings, then you can actually process this statistically in a way that when I tell you that we are talking about shape and morphology, we are talking about shape and morphology, because this statistical processing of the data removes all the variation that is due to size. So it's not anometric inference or size, although you can also get the data. It has nothing to do with the position of how you work with anything, and it has nothing to do with the orientation of your wing. So you're actually able to extract the vector shape in this particular trait, which means that once you have a vector, you can do all the beauty that you can do with multivariate stats. This is, for example, a principal component of wing shape in Drosophila. Every single dot that you see here is a fly, right? It's the wing of fly. And this is what we call a morphospace, right? So once you have a morphospace, it's basically telling you the phenotypic space that is explored by your population. And you can make that of inference on this, but this one is what we are going to talk about now. So if I take this particular fly and I average right and left, I can obtain a representation of a mean phenotype of this individual. So that's mean shape. If instead you take this fly here and you measure the distance from this fly to the average of your population, then you can get a measure of variability, right? It doesn't matter, this fly looks very different from this fly, but the distance to the average is the same. So they are equally variable. We don't care about the mean, we don't care about the shape, we care about how far away they are from this population. This is a metric of variability in your shape. And fluctuating asymmetry, which is fantastic, is because it's actually not depending on where you are, but it's actually measuring in the same individual kind of a replicate of a phenotype, and tells you how noisy this process was. So at the end, we have a metric for mean. If you think about mean expression, this will be mean morphology, and two metrics that are giving us a feeling for the variability and the noise in the process that creates this one. So what is the genetic architecture of variability compared to a baseline of mean, right? And again, this is just to remind you that all these traits have the same, it's the same individual, so it's the same genome. You are making inference exactly on individual level genotype phenotype maps. So what we did, because this is what we do, is to run GWAS on these particular phenotypes, right? So what Saudat did first was to run what we all do also, which is an additive GWAS, right? An additive GWAS means we are assuming that every position in the genome is affecting your phenotype independently, and the highly polygenic assumption of complex traits. You're going to have thousands of positions in your genome that making tiny contributions to your phenotype. So this is what we find for the mean shape. For example, this is known, people have done this before. We have tons of positions in the genome that independently, this is the statistical assumption, also biological maybe, that they contribute to shape, right? But then when we do these scans for noise or for variability, there's absolutely nothing. So there's no position in the genome, and I think the sample size for this analysis is good. We're not trying to make inference of 50, for example, and we say we don't find it because you don't have power. There's nothing. There's no position in the genome that is regulating variability or developmental noise, right? So Saudat was very sad because we didn't find the gene that we were thinking. Think about it. The interesting thing about this is everything I told you before in gene expression are additive scans. So for gene expression, there are tons of positions in the genome that on their own independently contributing to the amount of spread in this particular phenotype. But if you go to high on other things, you don't. Right? So there are no positions in the genome that regulate this, which is really interesting when you think about it, right? So when you read a little bit more about development and higher order phenotypes and how is me keeping us together, you really quickly start thinking about higher order interactions, right? So this has to be an emergent thing that is happening because you have geo-regulatory networks or developmental processes that have feedback loops and all these things that actually makes us be here and be robust to perturbations, right? So then the obvious thing was to run a epistatic scan, which is a GWAS that doesn't assume that your positions in the genome are independently, but assumes that they have to be interacting with something else to affect your phenotype, right? So when we do this epistatic scan, which is basically this computationally very demanding, so when we did solid pairwise scans, so you take this position in the genome and you test it against the whole genome, then the next one against the whole genome, the next one against the whole genome, until you find the interactions. And there you are. So the genetic basis of noise, developmental robustness, but also of variability is epistatic. So it does come from the interaction between things. There's no one locus that controls this, but it's actually these higher order interactions that are defining whether you're going to be more noisy or less noisy, right? The way you can think about it is this. This is a circus plot that shows you these results, where you see that if you have an A in the chromosome X, only matters if you have a C in chromosome 3 to regulate how variable, for example, you are going to be, right? This position in the chromosome alone doesn't do anything, but put it in interaction and then you're regulating noise. The cool thing about this also is that when you explore this at the network level, they are forming a network. So we have experimental evidence that these things that are interacting, these statistical tests, right? Like do these things. There's evidence that these guys act together to regulate whatever in this particular scenario variance. Yes.
The experiment showed versus home microarray.
How what?
Home microarray can predict this.
This particular one?
Yes.
So we don't find particular microRNA here, but for example, in gene expression, microRNAs are, you see the signal there. So we are now thinking about that. Like it has to, especially in the part that I showed you that things increase in variance transcriptome-wide, there must be a higher order mechanism that kind of regulates many things at the same time. So now we're thinking and testing whether microRNAs are actually doing a lot of this action. And in this one, we don't capture really the obvious things that you put in there. Okay, all good up to here. I just have a couple of minutes, so we may finish with just a quick note on things that we are thinking about right now. And we want you to leave this room thinking that we should start thinking about genotype-phenotype maps as probabilistic instead of deterministic, right? So again, as I showed you in the intro, this is a deterministic GP map, and you can imagine that we are focusing on displacements of the mean trait of your phenotype. And we have learned a lot of things out of having this approach in the way we map genotype-phenotype things. But what I show you today is that the idea of a probabilistic GP map is everywhere, if you want to look for it, right? A probabilistic GP map means that instead of mapping to the orange, I map to the cloud of phenotypes that are around this average. And if you happen to have a noisy genotype, the cloud of phenotypes that you can map to is bigger, right? And it's probabilistic because you don't know if you think about normal distributions, then if you throw the dice of A one time, you would realize this phenotype and be small, for example. If you throw it again in the next situation, that you can end up being a very tall individual. And this is just based on the probabilistic genotype-phenotype relationship. But as I showed you before, it seems to be very clear, not only in transcription, but also in higher order phenotypes, that mean and variability have distinct genetic architectures, which means that these two things are coexisting and giving us information that are complementary. If you think about means, this is the approach that you go for, and I showed you already that the inference you make on the genetic architecture is different. If you take this approach, trying to focus a little bit more on the variance, you get inference that seems to be distinct, because I guess it makes sense that noise and average values are regulated and evolving in a different way. So not only, I'm not saying that we should move here, but I'm saying that these two perspectives are giving you a fuller approach, a fuller perspective to what happens phenotypically speaking. So if we assume that these probabilistic GP maps are everywhere, and actually we are missing all this information when we don't do it, what June is doing as a postdoc in the lab is trying to develop new theoretical models that actually incorporate this regulation of variability. So it's adding stochasticity in the models that we normally use. So just for you to imagine a little example, if you think about classic, and again, of course, there are exceptions of population genetics models, you are trying to map a genotype to a particular phenotype, and there's no stochasticity in this realization, right? So you cannot really study the evolution of stochasticity because there's no genetic regulation of stochasticity. If you think about quangen, we have incorporated stochasticity in these models, but normally it doesn't depend on the genotype. So it is there, but it's equally distributed. If you don't have differential genetic regulation for it, then of course you cannot study this evolution, right? So what we're thinking a lot about is what happens not only with variability, which will be what you can do with a phenotype perspective model, but what happens with the alleles that regulate that variability? For example, you're running experimental evolution, right? Are these alleles that increase and decrease variance, they are tracking what is happening with the phenotypic? You need models that help you model these things. And this is what June is trying to do, is to develop these new models when you can map a discrete genotype, so you can track it in time. And you have them regulating not only the mean, but also the stochasticity of your value, which at the end renders a phenotype that is probabilistic. And this is, we are very excited about that because then you can actually address questions that are super interesting, like how does variability per se, we assume is a trait, how does it evolve on its own? But also how variability shapes evolutionary trajectories. If you imagine that you have a genotype space and you are navigating the space, but the phenotype space that you can reach is not one, but the cloud, how is this thing shaping where evolutionary trajectories are, for example, going? And those are things that we can expect to do. We have these models that are incorporated in this particular things. Okay, I'm done. Almost perfect timing. I just want to thank my lab, which is all these guys who do all the work, funding and collaborators, and I'm happy to take questions if you have any.
I have phenotypes without under which conditions?
Both.
Sugar?
This one is only, I show you just controlled results, but we expose them also to high sugar conditions.
Then I'm confused. You just told me, I asked that we don't expect much variation under controlled conditions. You only see them under the stress.
In gene expression, yes. In higher order phenotypes, it seems to be more interesting because you have tons of G by 2 in higher order phenotypes. You have common, so things that you can find in both, but you have a lot of G by in higher order phenotypes too, but not in the amount, in the proportions that you find in gene expression. So gene expression in that sense, I think it's just because it's just closer to what the genetic variation is affecting your phenotype, that is a really particular, hugely affected in stress. But higher order phenotypes are not, and I guess it makes sense because you have all these buffers and levels of buffering of noise or perturbation that you have in the organism. So it's not like gene expression in that sense, for sure. Which I guess kind of, I mean, if you think about it, kind of expected, I would say. Yeah.
Good, nice talk, very well explained. I had a question about your idea with the derived allele. Very interesting. I wonder if there's a way to account for frequencies also in this test, in the sense that I guess the things that you catch in GWAS are not the super rare, deleterious thing, and then just by biasing by things that can contribute to variance in the GWAS might be the high frequency derived alleles that might even be beneficial or less deleterious. So could you account for this frequency?
So for all the nodes that I show you, that is where we are making the bias inference on.
Yeah.
All this is frequency matching and sign matching everything that is significant. So that's a really, really long computation, I think, to try to make it right in a way that everything we're matching the node is frequency based exactly where you are seeing in the 1,000 times. So we believe that despite the whatever frequency can contribute to this bias, is not driving bias.
Okay, frequency is based on this, whatever, 12 individuals that you started the population. Do you have a better?
We make the inference of frequency on the whole population.
Yeah, but the population is based on a few isomers.
Yes, yeah.
So this may not be representative of the natural frequency.
I think it is close enough. So when you compare this to a natural population where the bias of errors is huge, this is basically cutting the data that you cannot map anyways. Because I did a comparison with, for example, the initial inbreds, and I think it's you are just removing all these things that you're losing by chance and all this data. But I will say this is pretty representative in allele frequencies in the distribution.
You bring up
you mean the identity?
All only singletons of an isophenol will occur at a relatively high
Higher. Yeah.
That's, but I don't know what it does. I'm just saying.
Yeah.
This.
I mean, that's the whole point of making this population is to try to make to make more parts of the genome mappable. But what I'm saying is that when you compare to the inbreds to the outbreds, the amount of singletons and rare things that are happening there, it's actually not as fantastic as we thought. All of them will increase frequency. So I think in general, this same distribution of things that are rare, you also lose them in the outbred. But I see your point. Like if you make the identity of the symptom.
Another question I have. Sorry.
Wait. But what you know with this one? Okay.
No. There are the.
Yeah.
I was curious about the experiment about sugar versus not sugar, and the fact that you find some SNPs that are relating to the modes of gene expression.
Right.
And so, if I understood correctly, the development time or the spread of the age of individual is different in the two different situations. So you didn't explain it.
Yeah, sorry, I didn't mention. Yeah.
But if it's not, so imagine there is just a SNP just influencing a little bit
The time.
the timing or whatever, then it would create all those effects without any relevance to what you're talking about.
Sorry that I didn't explain that. So all these are flies that have exactly the same age, collected at the same time, where we know, for example, if they eclosed yesterday, they are 7 days old, but they eclosed yesterday or tomorrow. So we collect them one day after and one day after. We also account for when they eclose. So we do know what's happening with them biologically in this, if you're thinking of the eclosion time, we know where they are. These are batches that are corrected in gene expression. But when we collect the data, all of them are 7 days old, females collected in the same time of the day to try to reduce this. So they are not different in age. It's just that for this experiment, we took the same populations and we ran the eclosion time just to show that they are slower. But
in age since hatching or since
hatching. Since hatching, yeah.
So that you cannot control those. So
yeah, no, but the age in the fly you counted when they are close, when they do metamorphosis and they become this adult, and all of them are exactly the same. Of course, what is happening in development is happening in development, but the age of the sample that you're collecting, based on how we calculate for adults, is all of them the same. But yeah, well, this variation could come from there. But the reason why it's not driving the signal, I mean, I'm sure there's variation there, is because we do know exactly when they close. So we collected thousands, many more than we actually processed, to be able to account for that. Like if you are the first closer versus if you are the late closer, if you are in the tails of this curve, that this is actually not driving your signal. Or this is like a posteriori accounted for. Or controlled for, we'll say. Yeah, okay.
I was wondering, I'm more on quantitative genetics for and I have a question about the replicability estimation when you account for individual variance. And so you have a variance that come from your population, like average variance, and you would get individual variance within this variance, the average variance. For the account them in like heritability measurements, is it increasing your global genetic variances? Is it not changing anything? Because for breeding application or in animals or plants can change a lot of things. You can control also the individual variance between individuals from the same.
Well, the thing here is because they are all outbred, we're making inference of all the basically heritability methods that influence on the mean value that these genotypes have. So you cannot do what you can do is, for example, estimate, and I don't have this at the top of my head now because with somebody else not in my lab who did it, is whether the heritability changes for the same genes when you are in high sugar, right? Because of course you are increasing phenotypic variance, and we are showing that this is genetically regulated. So in theory it should increase. If you're interested in this number, send me a message and I can ask for it. But that's the only thing you can do. Accounting for variance just by estimating environment-dependent heritabilities. But the way you do it, you can, you can.
Also, same question about it. Is your individual variance estimation not part of the classical G/E estimation we made before? And you probably showed that there was multiple level of G/E interacting with each other because you're adding another microenvironment effect with sugar compared to control. But you also show that between individuals in each condition, you also still have variability that will probably, for now, account in the residual part of the model, but it's also the genetic part. So you have like a G by E effect, in your estimation. And so
if you think your first E is high sugar and your second E is
the first
microenvironment, exactly, yeah. And I think that's always there, and we don't think of like that. We just don't model the second E that you are thinking that is this microenvironmental thing. Yeah. Which I think is cool when we progress a little bit more with the last part that I told you, because then so the microenvironmental, so this second E, you can directly estimate from the fluctuating asymmetry curve. Because then you have that metric at the individual level. But we haven't done that yet.
There is one question online from Arnaud Estrud. He's asking what would be the link, if any, between genetically determined trait variance and genetically determined trait plasticity? Is there a link between them?
Yeah.
Yeah.
I think these are two things that we should combine before working on plasticity. Which I find is that they might be different things and they might be very similar things, right? Like normally we think a lot about plasticity in general, about changing the mean when you have this reaction norms and you're moving somewhere else. But of course you have variance that is coming with this particular thing. And plasticity, in the way I imagine it, is this increasing variance, even if your mean doesn't really change. So I think this can be the same thing or different things. Or plasticity inferred from the mean could be a different thing that plasticity inferred from variance. But this to me is an example of plasticity, just that the focus of your phenotype is the variance instead of, if you were to do a reaction normal variance instead of reaction normal mean, for example. Yeah, so I think there's tons of many of these things that are overlapping, and then we just have to think about them together. I'm looking at you like it was your question. Okay. Okay. Yeah.
One question only. I missed the point. By the symmetrical width like that.
That's what I wanted to find. But it seems that unless the symmetry is very big, it doesn't really matter. And then reading a lot about how these wings are made, there seems to be a really tight control of the asymmetry in size. That might matter more. Like you need to have your area that is very similar. And then there are tons of compensatory things for size asymmetry during development that can result in slight shape asymmetry that are not functionally affecting size. But when it's very big, then you can see that. Yeah.
So how does it work at the molecular level, the transcriptional variability? I assume, or I would think that the variants that are being shown up or selected are the ones that can accommodate mismatches in microRNAs or transcriptional factor variation. Have you been able to show that association? Or?
No, so the thing here is that, for example, we find that these locus and the loci are all over. And there's not particular, I can tell you where they are not. So if you have genetic variation in coding regions for variants, they are really depleted significantly. You don't want to perturb, which is very interesting because we imagine who cares about something that regulates variance. So this seems to be just purifying selection on excellence, but not necessarily regulation. But they are depleted in regulatory in cis regulatory regions, which is super interesting because there are like two studies that have actually done them. We don't know much about molecular biology of variants, but there are two or so studies that have done that in using GFP in cells, and then you can mutate something and see what happens. They find that the bigger effects are coming when they mutate cis regulatory things. So my feeling is that that's why they are there, because you don't really want to go crazy. You want to go crazy a little bit. And then that's why probably you don't have these big effects there. So this is the only thing that we can see that is depleted in where these P EQTLs are. But more than that, exactly of how this actually works. The other thing that is interesting is they tend to be trans regulators. So we find tons of trans. We find some cis, but it's definitely biased. If you compare to EQTL, it's definitely biased towards trans, which is another molecular feature, let's say, of this regulation. Might come from this idea that you need to have, you don't want this big effect thing, but you need to have many things kind of regulated that. But yeah, that's the same.
I had a related question, but so maybe I missed the information, but the PDQTL, do they explain roughly equally the increase and decrease in variability?
Do you mean in per gene?
Yeah. I mean, when you showed the volcano plot where you had only a fraction where variability decreases, do you also identify the EQTL?
No, and that's why I think this idea of exploring, comparing to EQTL is brilliant. It actually tells you what your findings is real, right? So something that we find that I found fascinating, and that might explain why we have the evolutionary bias that we see again, is that when you have an EQTL, and you have this EQTL regulating 10 genes, it's explanatory. The same genetic variation increases or decreases expression. So A is high for gene 1 and A is low for gene B, right? Which makes sense, right? But when you find the EQTLs that affect 10 genes, the same allele tends to always be higher or low, which is really cool if you think of mechanistically. It's not that they are fine-tuning for this particular thing. It's like they are. For example, it could be like generate this mismatch with microRNAs that in general will affect everything. And this is super strong signal. They tend to be doing the same while the other ones drop. And that might explain why you have these different selection pressures. But they seem to be a totally different thing from the EQTL regulation.
Thank you very much. Thank you for the nice discussion of thought, and see you all next week for the next.
Thank you.
Voy a ver.